And the Raiders won in overtime.
Freddie, you were supposed to.
You guys are lame.
Yeah.
Woo! Good job, Raiders. Okay, so we are continuing on. We just did a quick review over all the cell functions that we've looked at before. We were looking at ribosomes. Cannot stress this enough. Ribosomes make proteins. That is an important concept. It's really, really important to the function and like just life of a cell. So we have our ribosomes, and we didn't draw any pictures of ribosomes, I don't believe. I'm going to jump in with the last thing that we had, and then we will hook it up with a nice sheet. So let's see. We had ribosomes, and this is the last thing that you have. You have some more stuff written underneath there. They make proteins. Okay. Beyond that, we also had what was known as the endoplasmic. Are ours capital in your notes?
Yeah.
Okay. The endoplasmic reticulum. Okay. Now, with this guy, there are two versions. First off, this is a membrane system. It has folds and stuff. You're going to see the word, if you read the book, I don't know how often you guys read, oftentimes you're going to see this word in biology. So like right now, the fish over at the hatchery that we have, they have bellies that have yolk still connected. They've hatched, but they're out. So if I said, hey, they still have yolk sacs connected, it would be yolk sac. When you see the word SAC, it's just like a regular bag, but it's in a biological sense. So like I get a sack at the store that's made of plastic. Anytime that I have some little region where I hold stuff within a cell or anything related to biology, it's SAC. Okay. Yep, that's the only difference. And I had to Google that because it's always bothered me. Anyway, the endoplasmic reticulum. This creates lipids and proteins. So membrane, system, and this is the last thing you guys have. It might be worded a little bit differently. Produces lipids and proteins. Now, a second ago, we hit on another idea. What did we say was the thing that comes out of the nucleus, specifically the nucleolus, that makes proteins? DNA. Not DNA. You're getting closer. What comes out of the nucleolus? What's the whole point of that? It does start with an R. The nucleolus produces something.
Oh, ribosomes.
Ribosomes. Ribosomes are what we've said make protein. All of a sudden, right next to this, we say, hey, the ER, remember that's what we're going to call it, endoplasmic reticulum is ER, it makes proteins. What do you think if I know that ribosomes have to make proteins and somehow in its function the ER makes proteins? What do you think we're going to find in the ER? Ribosomes will have to be there to make those proteins. So looking at it, the very last things, which one do we have first? Smooth or rough?
Rough.
Okay, the rough ER. What's ER again? Endoplasmic reticulum. The rough ER. This thing is going to be pretty clean to pick out. We're going to say this is where ribosomes attach. So within our endoplasmic reticulum, we do have two sections of it. There's a place where ribosomes attach, which by virtue of what we've just spoke about, what gets made in the rough ER then? Ribosomes attach there, and what do ribosomes make? Proteins. This is why we say the ER makes proteins, because it's where my ribosomes connect. Okay, beyond that, we have the rough ER where ribosomes attach, and I'll just say protein production. Production. And one step further, we have the rough ER. We also have the smooth ER. Okay, within our smooth ER, two big things that happen. First off, right off the bat, this is where we take energy and such to make our complex carbs. So within our reticulum, we can make proteins in the rough ER with the ribosomes, and we make carbohydrates. And I'm just going to say carbs for carbohydrates. Carbs and lipids. Now, remind me real quick, what's a lipid?
Fat.
Where have we seen lipids elsewhere in the cell? Weird. Maybe with a little bit of phosphorus mixed in. Phosphorus-based lipids. Check. That's okay. You don't have to remember it off the top of your head. Check. Between the four of you, I think we're going to find it. We're looking for lipids showing up anywhere in our cell. Maybe something big that we focused on. You're looking for the word phosphorus or a part of it in lipids.
Oh, phospholipids.
Oh my, phospholipids. What do they do?
They help
What part, where did you find phospholipids?
section. Or are you talking about like?
Where in the cell would you find them?
Oh.
I think there's a bilayer, if that helps anybody.
Yeah.
Where's the phospholipid bilayer? Somewhere in the cell. It's not an organelle. Not directly anyway. What separates our cell from the world around it? If it's a plant, it's a cell wall. What's right inside the cell wall?
Oh, the nucleus.
Not directly. Go back one section, and even before you turn your page, can you tell me what we talked about before we went inside the cell? We talked about the outside of it, right?
Oh,
What do we call the outside of it?
Nuclear.
Nope.
We talked about it.
We drew a bunch of pictures with things like, I'll even draw, here's a phospholipid.
Oh,
Here's another phospholipid.
The things that we
Oh,
throw
no,
out for water, like they have the protein transport.
Man,
for proteins.
so there are transport proteins that live out here and on top of it and carbs that join on. What am I drawing right now? This is the phospholipid bilayer of the what? What's the outer structure of our cell, not the cell wall? Weird. Phospholipid, that's where I was at. It's my plasma membrane. Check it out. Do you think it's possible that from time to time something happens where one of these gets broken?
Yeah.
Like naturally happens, right? We change the siding on our house, your skin cells fall off your body constantly. When it gets broken, if we don't replace it or fix it, it's pretty much death for that cell because now more stuff gets to get inside and it kills us. Where do you think these lipids that have phosphorus within them produced in the smoothie are might be useful?
For the cell.
This is the healing center, essentially, when we're looking at it from the lipid point of view. Okay, now beyond that,
So like that's kind of like, for example, if you want to run or something, that's kind of the science behind it. If you like sweat, you make it really hard.
can you, maybe, I didn't quite grasp that.
Like, say you go on a run, for example, and then like the next day you're already like worn out and stuff like that because you sweat. Obviously, you lost electrolytes, all that, but like using carbs, that's basically the science behind it.
So, kind of, I would say not at this level. The analogy that I would use, rather than like, you certainly do have to replace all those things, but it's outside of biology. Oh, we can do this. We can use biology. If you cut yourself, what does your body do? Like, if you're bleeding, how does it stop it?
starts wounding. It's like it starts.
It clots, I think is the word you're looking for, and then it forms a scab, right? Now, your blood makes it clots. It does something special. This is the same idea, though. This is like our fix-it guy. Cool. This is a handyman that's going to replace the siding on our house. That's the easier way to think about it. Okay, beyond this, the Smoothie R also detoxifies a lot of different substances. So if we get something in the cell that's toxic just naturally, and it depends on what it is as to whether it can deal with it, but if you get something bad in, the Smoothie R can help to take care of that a bit. Cool. All right. We are getting real close to this. We only have, I think, one more little section here.
Yeah.
The only thing we haven't looked at is how does one process, transport, and store molecules within a cell? Who was that? I think it's Fred, which worked out perfectly.
Oh yeah.
Okay. Do you still have your notes from the other day?
Yeah.
Sweet. So, last little section, and let's dig in.
Oh,
I can, and I will. Yep. I'll give you a minute. I'm going to write this and then I'll go. Okay, the last little section that we're going to deal with is processing, transport, and what?
Story.
and storing molecules. Now, this is an important piece of any structure that you're ever looking at. A city to function needs a place to process, to transport, and to store stuff for the city, right? Like, we have that here. A boat, anything that you've ever dealt with in your entire life has this system. Your house has a way to process stuff, to store it, and to move it. Like, every part of life, this is fundamental. Now, it's no different than within the cell. I'm going to give you a second to catch up. Are you all caught up now? Okay. Everybody feeling good?
Yeah.
Sweet. Here we go. Fred, what's the first thing that we're going to look at? That's how I learned to pronounce it. We're going to change that. The first piece of this process, transportation, weird, and storing is the Golgi apparatus. Golgi. The Golgi or Golgi apparatus. Now, I do have a worksheet for us, and I know that we have this one. The ones that I printed out for you the first time, the pictures that we have, we don't hit every single piece of it. Like, there's stuff that we just don't cover. It's going to happen no matter what class you take until you're at like the PhD level. But as we're looking at this, we are going to go ahead and pronounce this. How would you say this word? What? Where's the R? I see. That's my G.
Okay.
G-A-W-L. Well, gall, right? Gall. G. Golgi apparatus. Okay. As we're looking at this, I have a okay picture. Not the best, but it's all right. As we're looking at this, Fred, what's the Golgi apparatus do?
A flat end stack of membranes or membrane that distribute or package protein.
So we'll say, I'm going to change the way that was expressed, but that's perfect. Right on. Okay. As we're looking at the Golgi apparatus, these things, or this thing rather, it manages, stores, and packs proteins into sacks. And remember, this is without the K, it just means that we have biology. It's still a bag, essentially. Into sacks called, what were they called? Vesicles. And we'll hit vesicles here in a few minutes, okay? All right, we are going to see this word vesicles show up again in just a moment. Now, with this in mind, if we're trying to manage, store, and pack proteins, let's just think of like the assembly line. Ribosomes, which make proteins, were made in the nucleus. They get then sent from the nucleus to the Golgi to the endoplasmic reticulum, where they connect and they start making proteins. Now that we have all these proteins made, they need to get to the Golgi apparatus, where they'll then be packed into the specific tubes to transport where they need to go. So, as we're looking at the Golgi apparatus, a couple little comments that I have. Things that are dealt with in the Golgi apparatus come from the ER. Whether it's lipids or proteins, they come from the endoplasmic reticulum. If it were the ribosomes, they're going to come from the rough ER. If it were the lipids, they're going to come from the smooth ER. Okay, so we get things from the endoplasmic reticulum. And then a follow-up comment, and this is kind of going weird, vesicles, which we'll define here in just a moment. But there's a couple different ways that they act. They can fuse with the plasma membrane. And I'm just going to say plasmem. Now, these, when we're looking at these, we'll define it better here in a second, but I'm just going to say these are essentially tubes. Again, they're just more tubes that stuff moves through. Is it possible that I have a cell that makes a protein that a cell somewhere else needs? So in order to move it, then I have to take it out of the cell that made it and get it out into my blood to be able to be taken by another cell, right? Okay? That's what's happening with these vesicles. They can fuse with the plasma membrane to, I'm going to say is it excrete or excrete? With just a C? How sure are you? That looks right. To excrete proteins or move within cell. So maybe we have a protein that's been made that is kind of full circle. We had a ribosome made in the nucleolus, goes to the ER, makes a protein, gives it to the Golgi apparatus, and now maybe this new protein needs to be used by the nucleolus as well, so it can just ship it right back to help function there. Or move within cell. So my vesicles from this Golgi apparatus can spit stuff outside of the cell or inside of it. Okay, a lot going on here. Sweet. Man, there's so many pieces. So many pieces. All right. The good news is I think we only have 2 more.
Yeah.
Sweet. Kind of. Do you want a picture of the Golgi apparatus?
Sure. Yeah.
Do you want me to draw it or can I just show you?
There's all cool.
I'm going to actually use this one and we'll see if we can't find it in here. And that's not going to be upside down, so that's good. So let's check it out. We have a lovely cell living the life, doing its thing right there. Okay. We're just going to look at a few different little pieces. And we'll do this again and again, I'm sure. What's this?
Cytoplasm.
So the goo around it, that dark blue, is the cytoplasm.
Oh.
What about this thing? It looks like a pill with a bunch of little folds in it.
Oh yeah, I got that one. Cell structure. Oh, interesting.
I'll-
Cellulose?
It's not. So this is an animal cell, so we're not going to see. This is a mitochondria that I'm pointing at right now with my cursor. Okay. Notice here's a folded structure with a bunch of yellow dots called ribosomes. What is this big structure right here that we see? Yep, this is the whole piece is the endoplasmic reticulum. All of this out here is the rough ER that we see. And notice it's right next to the nucleus, which makes sense because when this RNA and ribosomes combine to make lipids and things, they come out here. Okay, so we have our endoplasmic reticulum is all these little folded ones here. What about this? This looks to me like a flat membrane that's folded. Gosh,
Oh, wait, Golgi apparatus.
that's my Golgi apparatus.
Oh.
Right there is a Golgi apparatus. Now, notice on this scale, we have the ribosomes that are immediately next to it. Why would it make sense that they're immediately next to it? What do ribosomes make? And what does the Golgi apparatus sort and transport? Naturally, it should be next door, right? Like, it'd be dumb if it were all the way on the other side of the cell. It wouldn't make sense. Okay. Now, the only other little pieces that we have to knock out have to do with vesicles. So I'm going to switch color here. Thoughts on what color I should use for vesicles? I'm apprehensive with purple only because that's typically what I used for the other one. I guess we're limited to colors. I'm going to use orange for no particular reason. I'll use white. All right. Just kidding. I really won't. Not funny. All right. Here we go. Last little section. We are going to define for ourselves vesicles now. Now, we've used them here, and I spoke it a moment ago. We didn't write it down. Anybody recall what a vesicle did? Or what do you think the shape would be if I know that it...
Can fuse with plasma membrane to excrete proteins or move within cells.
So if I want to pump a bunch of proteins through a cell or out of a cell, what does the structure that they move through look like, do you think?
Like tubes.
They're tubes. They are tubes, like a subway system, if you will, transport, whatever you want to think of it as. For these, we're just going to say these are tube structures, or totally tubular, if you will. Tubular structures. Whoa. That help, or that, yeah, I'm going to say help instead of facilitate. Move. And I'm just going to say materials. Now, I'm not going to write it here, but remember, they can move materials inside of a cell, or they can move materials to the outside of the cell. Okay, so we have our vesicles. Now, a few other things. We're not going to go into it like crazy right now, but they do store some stuff from time to time. They store food, essentially, not food like you and I eat, but any molecules that the bonds can be broken to get energy from. That's what I would call cell food. They store some of it. They are also made of a lipid bilayer. Now, I'm not going to make us look at them, not here anyway, but here's my picture just from my notes. Here's what I have for, notice there's a little tube here that stuff can move in, and it's just a lipid bilayer again. Cool. Smaller lipid bilayer than the plasma membrane, but same idea. There's my ER, or my Golgi apparatus. I didn't have color pencils. Okay, so kicking forward, we have some vesicles. Can you tell me any types of vesicles, Fred?
That I did not cover.
Okay, so one type is what we call a vacuole. This is just a type of vesicle. Now, specifically, a vacuole, most common in plants, mostly in plants, but there are vacuoles in some human cells, in animal cells. Okay, so mostly in plants. Beyond this, the vacuole, since it's just a tubular structure, it can hold stuff. Like there's a hollow inside. I'm going to say, and you said you do have it. What does this vacuole do, Fred?
It's used to store enzymes, food, and other materials.
Awesome. I'm going to go ahead and say holds enzymes, which this is a ways back. Can somebody remind me what an enzyme is? I have some in my mouth that help digestion start here. So you will find it in saliva. Bam, bam. What do we say is like the most important thing out of all these macro proteins? These are proteins that speed up reactions. So when we need to speed up a reaction to make sure we get the energy and time to use it before the cell dies, it has to use an enzyme. Okay, so they store these. Beyond that, there's only one more, holds enzymes and other materials, I suppose. Other materials. Do you have any other pieces, Fred?
Lysosomes.
Lysosome. Okay, notice this is also a type of vesicle because we're sub-layered here. These things, this is like the garbage man, is what this is. Which, what do I mean by that, Fred?
They digest excess or worn out organelles.
So vesicles that break down, I'm going to say that instead of digest, break down and remove worn out organelles from cell. I'm going to say worn out organelles and excess food. I'm going to put parentheses around food or quotations. Okay, now lysosomes. As we're looking at these, anywhere that you found them existing, Fred?
I didn't cover that part when I saw them.
Okay, so they digest organelles and extra food. Beyond that, I'm going to say also digest or break down things that might be bad for a cell. So like if you go swimming in a dirty pond and you have a cut, you might be worried about getting infected. What might infect you if you go swimming in dirty water? Bacteria, good. Anything else? Bacteria can make you sick, right?
Yeah.
What else can make you sick? So germs are a wider field. I'd say bacteria would count as a type of germ, for now at least. Gosh, it wasn't the COVID bacteria, was it?
Virus.
Virus.
Oh my god.
Okay, now there are other pieces. Some of these, I'm not going to talk about it in here. Do you remember what stops the tail ends of our phospholipid bilayer from sticking together so that stuff can still move through?
Like fats.
It's a type of fat. Cheerios say that like it's looked at as a bad type of fat, but it's not really. It's cholesterol. Love it. Nice job, Andrew. Okay, as we look at this, it turns out that there are some other structures that we didn't cover. One of them is called a peroxisome. I'm not even going to write this one down, but it's worth talking about. Okay, peroxisomes produce something very, very specific. What's the brown liquid, or the brown bottle that you might pour on a cut and it bubbles? You shouldn't pour it on a cut, but people do.
Alcohol.
It's not alcohol. That would hurt. It's hydrogen peroxide. H2O2 is what that chemical formula is.
You said like 2% of it is only of it, and then 90%.
Yep. You really shouldn't use it, but that's okay. Like it kills cells if there's enough of it, which if you're pouring it on your body, there's enough of it to kill cells. Okay. With that in mind, what do you think peroxisomes might make within a cell?
Peroxisome.
Mm-hmm. Knowing that Fred just correctly identified that liquid as hydrogen peroxide, what do you think a peroxisome might make?
The opposite. Because what it makes.
Similar.
Can I just say peroxides?
Yeah.
Like peroxisome, it makes peroxide. So hydrogen peroxide at big quantities is terrible for us. Turns out, though, that you have to be able to produce it and to eliminate it from your body because peroxide is used to make fatty acids. So when we have fatty acids via lipids and things, we need peroxide within us. Now, for this, I'm not going to write this next part down, but it's worth talking about. There is a huge amount of teamwork that has to happen within our cells, right? And all of this information that tells every little piece what to do and how to do it, what's the information?
DNA.
It's our DNA, which is stored where?
Nucleolus.
The nucleus in the chromatin specifically. But like, if we just look at any little piece, so last time I'm going to make this comment, most important macromolecule from my perspective. Now, whether that's really true or not, I don't know, but proteins are really important. Okay, if we look at a protein, let's just think about the craziness it has to function within our cell just for that one protein to get made. First off, you have directions in the nucleus, right? That's your chromatin, your DNA. It says, hey, here's how you do it. Once you have that, you have to then put DNA together with RNA, which is another type of genetic material, and they, in the nucleolus, combine to make a what? What makes proteins?
Oh, ribosomes.
The ribosomes. Now, again, just check out just for a singular protein what's happened. Directions are in the DNA. It fuses with RNA and other stuff to make a ribosome. The ribosome then goes where? Not yet. We don't need ATP here. Check your stuff. We've looked at all these today. Where do the ribosomes land?
Oh, in the fossil lipid.
Why? Where'd that come from, Fred?
Because you said something about earlier how like they needed to structure, like has something to do with the structure. Like,
So the vacuoles will have that bilayer, yes. So I see what you're getting at. So we have ribosomes made here. And then through microtubules or maybe some vesicles, they move to the rough ER. So here's my ribosomes, and they make proteins, right? And they make proteins, and then they ship them where? To the Golgi apparatus. Now, we have these proteins packed and ready to go. They then do what? Where might they go? They travel through vesicles. Is it possible it spits out of the cell and we now have a protein floating around for another cell? Or instead of moving it out of the cell, where else can we move it? Inside the cell. Okay. We needed like 5 different structures just now without really talking about the energy needed to carry out all this that's made in the mitochondria. And like all these pieces, they work together. It is incredibly complex, but we're incredibly done with this for now. So, hit it.
What's so spiky stuff out on the blue?
Are you looking here?
No, like on the outer shell. Yeah.
Ooh, what do you think that is? Small
Oh.
projections outside of this guy.
Protein transporters?
Maybe. I think there's another one. Little projections that come off of a cell.
No projections that come out.
Like little fingers or hair-like things. Maybe they reach out and grab oxygen, or they push stuff through our body, through our digestive tract. That's a cilia. Yep, 100%. There is no flagella or flagellum on this one. The flagellum would be longer. Cool. All right. So here's where we're at. I'm going to put something up on there as well. I will post this to the Google Classroom. You guys have this in your notes as well. But I'm going to put this here. And then let's see if I can get it. I'll get you guys some copies, and I'll pause it awkwardly here. From the handout that's on the classroom, the Table 1 Summary of Cell Structures, it's in your book. We're going to make a Venn diagram. What's a Venn diagram?
Were you able to see and label the things of something?
So if I wanted to draw a Venn diagram, how do I start? I need a circle. Now, I think, and I'm going to switch colors here. I don't know what color's appropriate for each. I'm going to make a big circle. And I'm okay if you guys do this in your notes or on the back, but we have 3 types of cells that we've possibly discussed. We have really complex cells, whether it's plant or what's the other one? animal. So check it out. We have some animal cells. Okay, and then using this lovely green color, I also have what other kind of cell? We have plant cells, and there's really one more that hangs out here.
Neutral.
What if it's not a plant or an animal?
Oh, mix.
Ooh, I don't think we can mix them.
Neutral.
What do you mean neutral?
They both
Oh my gosh.
common.
Which, are these cells complex, plants and animals?
Yes.
They're very complex, right?
Yeah.
What's the non-complex cells? You know it.
So, DNA.
You're pros at this point.
Uh,
You're pros. Okay.
prokaryotic.
Hey, there we go. We got some prokaryotes. Boom. You guys have on your page a big ol' list of the cell structures and organisms, pieces that we looked at. Now, we discussed every single one of these. I will tell you straight up, I am going to ask you to be able to tell me what something does, especially if it's one that we talked about a lot. What I want you guys to do now as a group, but I want you each to have it, I want you to find me the structures and place them in this Venn diagram. If you need to use numbers, I'm trying to decide, depending on how big you wrote it, some of these words are big. If you want to physically put the stuff, like I know, for example, that in here we get chloroplasts. I know those are only found in plant cells.
Cell was also found in plant.
Hey, cell wall, but I think cell wall might be found
Oh, so
elsewhere.
it would be a prokaryote.
So, love it, because you're using this sheet, right? Okay. As we look at this, let's look at the first one. We'll look at this one right off the bat. Plant cells, sorry. Chloroplasts are in plants. I skipped ahead. Cell walls. They're in plant cells, fungi, and some prokaryotes. Where on earth would I say is a region that has prokaryotes and plants? And you don't write these numbers, but I'm going to just so we can talk about it.
4, 5. Oh wait, 4. Yeah,
4 is the overlap between prokaryotes and plants. That means that at number 4 then, we should get cell walls. Okay, I want you guys, as a class, you can work this out. You might work independently. It's entirely up to you. I want you to do this for all, and I'm going to number mine. I'm just going to go down the list on mine just so you know and number them 1 through whatever. I want you guys to construct this Venn diagram. Once you have it, we're going to do models tomorrow, but I will put this up on the board. Once you get it, I want you to knock out for me this evening. Let me move over to the other side, and I hate where it's at. We'll just do it right here. I also want you on a separate sheet, or in your notes if you'd rather, we're going to knock out homework 7.4, which you will find waiting for you on page 192. And we're going to do 1 through 5. Now, if you're traveling, I'm going to hook it up. In here, I'm just going to put the pictures up on the board. So at the start of class tomorrow, here is the assignment. Now I will copy and paste that just on there so that people on the video can see it. Page 192, 1 through 5. Excuse me. And I know that 6 is showing there, but we don't need it. Okay. By tomorrow, I want you guys to have Venn diagram completed, which I'm going to cover it for now. I want you to have Venn diagram completed and these 5 questions. Cool. Play-Doh cell structure. I'm going to see if I can't find something cooler than Play-Doh to use. I will try my best to get it set up to do a bunch of microscope stuff in the coming days as well. Cool. All right, I'm going to move this out of the way. If you're looking at this, pause it and take a screenshot now because it's going to disappear in 3, 2, 1. Cool. All right, we get 9 minutes to finish this up. We will check this out tomorrow.